Um, I'm Kavita, and today we are going to discuss canal system in sponges. In our previous class, we have discussed about their general features as well as classification. So while discussing the general features of Porifera, I said canal system is one of the very important characteristic feature of all the sponges. So in today's class, we are going to understand, or at least try to understand, the working mechanism of this canal system and also its significance. The learning objective of this chapter are to know the canal system of sponges, to explore the types of canal system in different classes of sponges, and to understand the functions of canal system. By the end of the session, you will have a clear-cut idea of the canal system of sponges and its significance too. Now, what is a canal system? So, the canal system is a system of intercommunicating cavities that are bathed by the current of water that aids the sponges to carry out vital life processes. As you all know, the sponges are simple forms of metazoans or multicellular animals with the least specialization of any part. So that means they don't have special systems for digestion, for respiration, excretion, circulation, nothing. So here, but still they need to survive since they have aggregates of cells. And this challenge of surviving has been overcome by adopting a special kind of water system, okay, called as canal system. You find this kind of water system in one more phylum, that is echinodermata. There it is called as water vascular system. Here it is called as canal system. Even though slightly they are similar and particularly in functions, but the system has a different structure. In case of Porifera, it is just a establishing system, okay, and it is a system of canals. That's why the canal system, cavities, okay, and some specialized cells that help in the movement of water, creating water current, some inlets and some outlets. So usually water canal system will have, a pipeline will have, there will be inlet, there will be outlet, and there will be pipes, interconnections, etc., through which the water moves in, then goes out. Okay, in case of sponges, this canal system serves that purpose. So water enters into the body of the sponge through this specialized canal system, okay, and then it gets circulated within that cavity called as spongocoel, and once its function, its main purpose, that is helping in respiration, bringing the food, okay, and helping in excretion, everything is done, the same water will be thrown out. If you recall, when we were discussing about general features of Porifera, we said that the one of the important feature, the name Porifera, is given because it has a numerous pores, okay, and those numerous pores on the body wall are called as ostia. We also said that there is a larger opening called as osculum, and through the ostia water enters, and through the osculum water exits. So in between this ostia and osculum, lots of things happen, okay, and that is what canal system is. So the entry of water through ostia and exit through osculum. In between, respiration, excretion, circulation, digestion, everything takes place. And this particular canal system, it is not the same in all the members of sponges. It is simple in one form and it is very highly complex in other form. So again, evolution, okay. So slowly in different groups of the sponges, the canal system has evolved in different ways. So according to the complexity of these canal systems, or the order of increasing complexity, we have 3 plus 1, okay. Basically 3 types of canal system: asconoid, cyclonoid, leuconoid. So we are going in series, okay, of complexity. Asconoid is simple type. Modification of asconoid is a cyclonoid, Leuconide type is highly evolved type of canal system. There is another type, Ragon type, which is not an independent type, rather one type in Leucon type itself. Based on the derivation, it gets a name as Ragon type. So in this chapter, we will be discussing all these 4 types of canal systems found in case of sponges. Let's see one by one. The first one is ascenoid canal system. Now, this type of canal system, it's very simple form of, one of the simplest form of canal system found in sponges. Okay, and it is commonly found in Leucosolania, okay, one of the calcareous sponges. Now, here, so usually the body is vase-like, and what happens? You see, you're all familiar with these pores, isn't it? These are ostia. Okay, and the ostia, we know it is nothing but a intracellular space in a porocyte. A porocyte is a special type of cell which has a central canal-like structure through which the water passes through. And these ostia, this is one porocyte actually. So through this the water will enter, okay, it will enter into the spongiocele. So the beginning point is ostia, passes through the pinacoderm, then the mesenchyme, and then the coenoderm, okay, single passage, and directly from there it goes to the spongiocele. The spongiocele is lined by special cells called as choanocytes or collar cells, isn't it? So we also know that these choanocytes are provided with the flagella, and the beating of these flagellum will help in creating a water current here. So water flows, okay, in a circular direction for some time. So water current is produced here, and now what happens? What is the use of it? The water that is coming in, okay, the water that is coming in through this, it is bringing, okay, water, dissolved oxygen is present, okay, and then some minute food particles will be present. So when the water circulates here respiration takes place. Oxygen is taken in by these cells, then carbon dioxide is released to this water, okay, and then the food particles will be taken in by the choanocytes, and it will be transferred to the amoebocytes of the mesenchymal area. Plus, what else happens? Simultaneously, these cells, okay, are going to release the excretory product to the water. So excretory product is thrown out, carbon dioxide is thrown out, okay, food is taken in, okay, then oxygen is taken in. So everything takes place within this process, within this canal system. Now once all the waste materials are released to this water, okay, once it is released to this water, now what happens? This water is thrown out through a opening, and the opening is called as osculum. Okay, so ostia is the entry point of water, then osculum is the exit, okay, of the water. In between these two processes, all the physiological activity will be carried out. So here the path is very simple from ostia, okay. So here I think this can be much easier one to understand here. So let me zoom it so that you can have a clear idea. So here we have the ostia, the entry point, passing through this porocyte, coming to the spongiocele and getting circulated here, and then being thrown out of the osculum. Okay, so this is the passage of canal system in, or passage of course of water, you can say, in case of ascenoid canal system. One of the simplest form of canal systems, okay, and it is found in Leucosolania type of Leucosolania sponge. It's one type of sponge anyway, calcareous sponge. Hope you could recall. Okay, so here choanocytes play a very important role in creating the water current. Okay, so that is about the ascenoid type. I'll just repeat with this flow chart here. So water enters through ostia, enters to spongiocele, then leaves to osculum. So 3 components are involved in this simplest form of canal system. Next one is cycanoid canal system, cycanoid type of canal system. So it is commonly found in Skypha, okay, and here it is a modification of Ascan type. So in Ascan type of canal system, so Austria is directly involved. Through that, water passes and comes to the spongiocele. But here what has happened is, see, in case of Ascan type, the body wall is straight. But what has happened, and here so that means there are the pinacoderm and the choanodem. Choanodem is present. Okay, the same thing here. What has happened? So let me just draw it here. Okay, so let me take color pen. Yeah, so this is the ascenoid type. The same body wall. In case of cycanoid type, it is folded. It is folded. So what happens? See, this side, it is the choanodermal area, this is the pinacodermal area. Okay, so now you see choanodermal area is folded inward like this, pinacodermal area is present outside. Now that means this is a one canal, this is another canal, both having a dead end. How the water moves in and out? Very simple. Let's see it here. So let me take this picture and have a closer one. See, this is the, actually you can see the folding, okay, the folding of the body wall. See, the red line is the pinacoderm and the purple one mesenchyme, the inner line is the choanoderm. Now here there is a entry of water. So these are there is a canal here, okay, there is a canal that is called as a incurrent canal, and then there is another canal called as a radial canal. This canal is called as radial canal. Similarly, you can see in all the ways. So what you special you find is that radial canals are lined by the choanocytes, flagellated cells. So it is called as a flagellated canal. Whereas these incurrent canals are lined by pinacocytes. There is no flagellated cell. Now the water enters to the incurrent canal through dermal ostia. Okay, and from there it comes to the this one in the incurrent canal, and from here it goes to the radial canal through the these prosopiles. Okay, they go through the prosopiles. Now here you can see the prosopiles. These are prosopiles. So I think I can just use another picture. Yes, this is much easier to understand. Again, alternative one anyway. See, here you can see the direction also. So this one, yes. So here you see the dermal ostia. This opening is the dermal ostia. Water enters to this incurrent canal, and from here you can see the passages. So they are called as prosopiles, and they come to the radial canal. From the radial canal it enters into the spongiocele, and that opening is called, okay, this opening, that opening is called as apopiles. Okay, it is called apopile. And then water comes to the spongiocele, just like in case of ascanoid type, it gets circulated. Okay, respiration, excretion, digestion, I mean feeding, everything takes place within that water, flowing water, and then the through the osculum water is thrown out of the body. So here the direction or the course of water is, first the outside water, surrounding water, passes through the dermal ostia, enters into the internal canals. From internal canal it enters into the radial canal through prosopiles, and from radial canal, which is also lined by flagellated cell, the water comes to the spongiocele through the apopiles, and through the spongiocele it goes out, okay, through the osculum. So again, ostia and osculum are inlets and outlets of water. There is no change. What has changed is the body wall, it is now folded. The straight body wall is folded and it has become more complex in case of cycanoid type of canal system, and this is found in case of cycon, okay, and Cysetta as well as in Gruntia. Gruntia shows a bit more complexity of a cycon type, where there is a cortical area and subdermal spaces are also present. Okay. The third type of canal system is Leucon type of or leuconoid type of canal system. So here this is basically seen in case of members of Demospongiae like Spongilla. So highly complex form of canal system it is, and its derivation is, it's not that in most of the cases even though like Sycon type is derived from Ascon type, but during the development of Leucon type you won't see Ascon and Sycon type. It is more complex one, direct derivation into Leucon type itself. So when you see a Sycon type of canal system, the embryonic stages they'll be having Ascon type itself, then they undergo modification, but Leucon type is not that way. So this is like it is a more complex type and doesn't pass through the Sycon and Ascon type of canal systems during the development. Now here, what kind of complexity is seen? Again, it is an incurrent of water and exit of water. In between that, the complexity occurs so in the structures. So remember the dermal ostrea there, then there is an incurrent canal, okay, then there is a radial canal, and then it was going to spongiocele. Here what has happened, this passage has become little narrower and more complex. Let's see what are they. So here we have the picture, okay, and I'm going to use my color pen so that will be much more clear. So there is a dermal ostia, so the water enters through the dermal ostia, okay. From the dermal ostia it should come to the incurrent canal. See, this canal, it is narrower, it was broader in case of a Sycon type. Here it is narrow tube like, okay, and then from the incurrent canal it goes to radial canal there, okay. Here instead of radial canal it is called as flagellated chamber. It is no more a canal, it is a chamber. It is almost spherical in nature, okay. It is more like a spherical structure, small as well as spherical in structure, again lined by choanocytes, okay, just like the radial canal. Here too they are lined by choanocytes. There it was canal like, passage like, here it is chamber like. So from the through the dermal ostia water comes to the incurrent canal, and from there it goes to the flagellated chamber. Now this entry is again called as prosopile. There too it was called as prosopile, here too it is called as prosopile only, okay. Change is only in the structure of the canal. Next, from this chamber, okay, the water moves to the next canal called as excurrent canal. You can see here excurrent canal, incurrent canal, and then the excurrent canal. And at this point where the water comes out of the flagellated chamber into the incurrent canal, okay, that is called as apopyle. It's called apopyle. So prosopile and apopyle same, okay, terms are same, but only the passage is different here. In case of these syconoid type, what has happened, radial canal open into spongiocele through the apopiles, but in case of these leuconoid type, from the flagellated chamber it opens into excurrent canal through the apopiles. That is the difference, okay. So it comes to the excurrent canal, spongiocele is almost reduced, okay, see it is almost reduced here, and the water passes through this and goes out of the body through osculum, okay. And this is the passage of water or course of water in case of a leuconoid type of canal system. I'll repeat. So in case of leuconoid type, which is highly complex, instead of flagellated canals or radial canals, there are chambers. Multiple chambers are present, and these chambers are more spherical in nature, and they are smaller, so they are restricted. Choanocytes are restricted only to this flagellated chamber. You won't find them anywhere else. That means what was happening in the spongiocele of asconoid type is actually happening in the flagellated chambers of leuconoid type, okay. And it is in multiple structures. Spongiocele was a simple sac-like structure with the reduced surface area. But in case of leuconoid type, because there are small chambers, the surface area has now actually increased, okay. And here the course of water is outside, I mean the outside water enters into the body first through the dermal ostia, it enters into incurrent canals. From there, it is passed to the flagellated chamber through prosopiles. Okay, and from the flagellated chamber, it goes to the excurrent canal through apopyle. Pyle refers to opening. So, excurrent canal, and from excurrent canal, it passes to the outside through osculum. Okay, so this is the course of water in case of a leuconoid canal system, generally found in case of demospongia. Now, among these, okay, there are some modifications. Some modification, that means some complexity is seen, okay, in different types of leucan type of canal systems. And this modification, it is again successive grade, okay, in the evolutionary pattern. The basic type is a Euryphilus type of canal system, leuconoid canal system, and it is the simplest leucon type. Okay, simplest leucon type. And here I think I can just focus it here. Okay, yes, this is the Euryphilus type of canal system, leuconoid type of canal system. So here you see the flagellated chambers, they'll directly connect with the excurrent canals through the large apopyles. Okay, apopyles are much larger here. So you can see the apopyles, direct chambers are opening directly through the large apopyles in case of Euryphilus type of leuconoid canal system. Whereas in case of apodal type, so that, yeah, here the apopyle is no longer a broad opening. Instead, it has become canal like. Okay, it has become narrow canal. Now this narrow canal is called as aphodus. It is called as aphodus type of canal. Okay, instead of apopyle. And when you go to the next one, the diplodal type, diplo refers to two, both prosophile and apopyle are canal like. Okay, in case of Euryphilus type, there is a broad apopyle, okay, directly opening, no narrowing, no tube, nothing, just opening. Whereas in apodus type, the apopyle has become narrow and canal like, tube like. Okay, so it is now it won't be called as apopyle, instead it is called as aphodus. And in the third type, diplodal type, what has happened here? Both the prosopyle and apopyle have become narrow and tube like. So this apopyle is called as aphodus. So you can see here, and prosopyle is called as prosodus. See, it is called prosodus. Okay, so since both of them are canal like, it is diplodal type. Okay, so in leucan type you find three basic types, modifications, evolutionary modifications: Euryphilus, aphodal, and diplodal type of canal system, leucon type of canal system. Euryphilus is found in Placina, aphodal type is found in Geodia, and diplodal type is found in Spongilla. Okay, so that's about the leuconoid type of canal system. Now, in addition to these three types of canal system, there is another type called as Ragon type. Now, instead of saying another type, it is a modification rather. In case of Demospongia, okay, the common bath sponge group, here leuconoid canal system, it derives from one of the larval forms called as Ragon larva. Okay, so in this case, in case of Ragon larva, this is found in larval form only, not found in adult form, but its modified form is found in adult leuconoid types. Okay, so here the spongocele is almost like triangular in shape, and flagellated chambers are present on these lateral walls. Okay, on the lateral wall, so called as spongosphere. Now here the broad flagellated chambers are present with the broader prosophiles and the apopyles. Okay, so and then it leads to a large spongocele, large spongocele. If you remember, leucon type is without generally with a reduced spongocele. In case of Regon larva, spongocele is much quite bigger, and here prosopiles and apopiles are also much larger, and the mesenchyme is much thicker. Okay, and here in case of, so this is actually found in case of larva. The similar type is found in adult leuconoid sponges. Okay, and here see, yes, now there is a subdermal space. Okay, so there is a dermal pore and the water directly doesn't enter into these internal canals. Okay, instead they come to a chamber. The chamber is called subdermal cavity, and from those subdermal cavities, so the water passes through the incurrent canal, which here is called as prosodus, canal like, see, canal like. Then it comes to the chamber, okay, flagellated chamber, and from there through the excurrent canal, canal like aphodus, okay, it moves to the spongocele. It moves to the spongocele. From the basic leuconoid type, there you want to find a subdermal cavity, but in Regon type you find a subdermal cavity in the mesenchymal area. Mesenchymal area has become much thicker. Okay, since it has similarity with the, or it is derived from the larval form, the Regon larval form, this type of canal system is called as Regon type. It is actually a leuconoid type of canal system which is derived from Regon type of canal system or Regon larva kind of canal system. So it is called as Regon type of canal system. But structures are almost similar with aphodus, prosodus, etc. Addition is that there is a subdermal cavity. Okay, so that's all about the canal system. 3 plus 1, 4 types of canal systems are present. Depending upon the complexity, the ascon is the most simple type, leuconoid is the highly complex type of canal system. What are the uses of canal system? So it helps in nutrition. Already I said the food will be brought by the water, incoming water. So that helps in nutrition. It also helps in respiration. Oxygen and carbon dioxide are exchanged between the cells and the water, water which is present in the spongocele in case of ascon type, or the water that is present in the flagellated chamber in case of other two types, flagellated canal and chamber in other two types. So direct diffusion takes place. Excretion, so waste material is also thrown out through this water that is flowing through the canal system. It also helps in reproduction because it helps in bringing the spermatozoon from other sponges. We know that sponges, even though they are hermaphrodites, cross fertilization is the rule. So spermatozoons are brought by the incurrent water, okay, and then the fertilization it takes place, and then the larval forms are also released to the water. So in that way, canal system of sponges help in reproduction too. And that's all for today's class. Let's check how much you have understood. Your first question is type of canal system found in Leucosolenia. Ascon type, Sycon type, Leucon type or Regon type? Answer is Ascon type, simplest form of canal system. Second question, Sycon type of canal system has, okay, answer flagellated canals, prosopiles, folded walls, all the above. Do they have flagellated canal or prosopiles or folded walls or all the three? Answer is D, all the above. All the three features are found in case of Sycon type of canal system. Third one, Euryphilus type is variation of Ascon type, Sycon type, Leucon type, none of the above. Euryphilus type. Okay. Option is C, Leucon type. So Leucon type is one of the modification of, Euryphilus is one of the modification of Leucon type of a canal system. Regon type of canal system is found in the members of Calcarea, Demospongia, Hexactinellida, none of the above. And the answer is Demospongia. So Rhagon type is the modification. Okay, the Leucon type which is derived from Rhagon larva is called Rhagon type. It's common in case of Demospongia. The last question is opening of the radial canal into spongiocele in Scypha. Okay, recall what kind of canal system is found in Scypha. So opening of the radial canal into spongiocele is apopyle, prosopyle, micropyle, dermal ostium. Answer is apopyle. Prosopyle is from incurrent canal to the radial canal. Okay, so that completes our topic of today. Let's summarize the class. Canal system is a system of intercommunicating cavities bathed by the water current. Okay, hope you are clear with the idea now. And there are different types of canal systems like asconoid, syconoid, leuconoid and Rhagon type. Canal systems are basically involved in nutrition, respiration, excretion and reproduction. And these are my references for the class. Thank you.